Alright guys, so today we'll be talking about the neoplasia. So this is going to be the introductions of the cancer cells, and on top of that we'll talk about the nomenclatures and also the different compositions of the cancer cells. Alright, so before we go and talk about in detail about neoplasia, let's define what neoplasia basically means, right? So let's go with that. So remember, whenever we say neoplasia, remember the name itself tells you something, all right? Remember this, if we break this word, neo basically means new, right? And the plasia basically referring as new growth. This is literally neoplasia means new growth. But remember, not every new growth is a cancer or neoplastic, all right? So then we have to define what is actually neoplasia basically means, right? So we basically define as this. At least Harrison, this is how Harrison defines it. It basically says that it is a unregulated, let's just say this, it is an unregulated cell divisions, all right? Unregulated cell divisions A. It evade apoptosis, right? It has a characteristics of invasions, all right? It has a character and can also metastasize, all right? So usually, whenever we say neoplasia, even though it is a new growth, but you have to remember that not every new growth is neoplastic or neoplasia, okay? In order to call neoplasia, it needs to have unregulated or uncoordinated cell divisions, right? And we'll elaborate more about what that means. And remember, it also needs to evade apoptosis, okay? It cannot die, and this neoplastic cells also has the characteristics of invading or going to different tissues and also metastasizing property. That's what they have, right? Now, what is this concept, this neoplasia basically means, right? Remember, another name for neoplasia nowadays, we said neoplasia directly equal to what? Tumor, okay? Or sometimes, we also call this in basic, we say cancer, right? And why we call them a cancer? If I give you a little bit of, all of this means same thing, right? The cancer is this word, basically. This cancer is actually came from the Greek word, all right? And Greek word, something called karkinos, all right? A little bit telling you about this. And also Latin word called cancrum, all right? So usually, all right, this cancer actually arises from this Greek word called karkinos, or also the Latin word called cancrum. But what does this mean though? It is basically referring as a crab. Cancer is basically referring as a crab. Okay, C-R-A-B, crab. And what does crab basically means? Remember how crab, they have their different legs, right? And when they actually invade, or let's just say when they're actually in one space, they don't, no matter how much you try to remove it, they don't usually get removed from that area, right? And the cancer cells are also like that. So that is why, this word cancer, that originated from these two different words, and which basically means crab, all right? Now, after this, right, let's talk about in little detail about what this means, right? But we'll do this more in a diagram, right? So if you look at this is let's say one cell, right? This is one cell. And what normally happens is that if you look at the one cell, it has a very beautiful circular, normal structures, okay? We'll talk about what is normal first. It is a normal cells. It has a different cells, different nucleus, right? And it has a solid architectures of the cells, right? And whenever, let's just say cell, example, let's say whenever cell proliferate, or let's say multiply, okay? What happens is that they need to go to a process of cellular divisions, right? They need to go to a process of mitosis. That's what happens. Now, before we go there, let me quickly talk about some stuff to you guys, just to remind you guys. Remember how we have in our body, right? We have a cell cycle, right? Let's just set the cell cycle right here. In a cell cycle, we basically have, what is this? We can simply say that, let's just say that we can simply say you have a G1 state, right? Then you have S phase, and you have a G2 phase, and then you have a basically, this is a normal cell cycle, okay? And you basically have a M phase, right? This is what usually happens. And then you also have a Z0 phase, correct? Now, usually the cells which are in G0 phase, these cells, even with the stimulations, right, they don't usually go a process of, cell divisions. And what are the examples of G0 cells? State. These cells, which are in G0 are basically neurons, right? You're talking about neurons. Not only neurons, you have, let's just say, skeletal muscles, right? Red blood cells, platelets, right? And obviously cardiac cells, right? So these are in G0. Now, you have a cells which are in G1 phase also, right? G1 phase. And remember, these cells which are in G0, they are non-dividing cells, all right? But there's also cells which can divide, which can actually, which are usually without stimulations, they remain in a G0 state, but which are stable cells, let's just say that. But then when they get stimulations or certain growth factor, they can actually move to G1 phase, okay? And what are these cells which are in basically in G1 phase? These are quiescent cells, right? And these ones are usually like if you're talking about, let's just say, proximal convolutive cells, right? Or let's just say liver cells. Liver cells are very important examples, right? Proximal convolutive cells, you have liver cells, right? Even some pancreatic cells, right? Because with the growth factors, they have a what? They have a tendency to actually go to a G1 state, and they can do what? They can rapidly multiply, right? Or not only this PCT, liver cell, pancreatic cells, you also have lymphocytes, right? Now, these cells right here, this one's actually, usually they're in G0 state, right? But with the stimulations, they can go to G1 phase. Now let's just talk about cells which are actually in a G1 phase only. What are the cells that are actually in G1 phase? Which means that they're always multiplying, okay? And there's importance of this when I'm doing this, right? The cells which are in like G1 phase, very, very importantly, your GIT epithelium, gut epithelium, let's just say that. These are very, very important, right? Because these are labile cells, right? Not only this, your skin, right? Cells in your skin. Third, like your hair follicles, let's just say. Or let's just say bone marrow, right? Bone marrow. Look, your gut epithelium, your skin, your hair follicles, your bone marrow, right? These cells are always like what? Rapidly multiplying, right? Like for example, look at this hair follicles, right? Our hair is always growing, right? So normally, so what happens is that these are rapidly multiplying cells, meaning that when cells has to multiply, they need to go through what? They need to go through something called cell divisions, right? Now, the hallmark of the cancer is that usually cancer cells arises from these cells that are actually in what? They're actually in this group, which is G1 phase, which are cells that are actually going through a cell divisions all the time, because the more you actually multiply and go through the process of cellular divisions, there is a more tendency of gaining mutations, right? And the mutations, like genetic mutations or genetic alteration, is almost always the cause of the cancer cells. Or always, like almost always, like, so you have to remember this, genetic alterations. This is very, very important. Usually in the cancer cells, it's a genetic instability, or let's just say alterations. It's almost always a, cause of, inducing a what? Inducing neoplastic, or causing or transforming, let's just say the normal function cell into what? Or neoplastic cells. So this is something that you have to always remember this, right? Now, so basically, look, and the cancer cells usually always are in the G1 phase, right? That's the reason why, like, usually you don't see any cancer that has to do with the neurons, right? Or let's just say, or cardiac cells, right? Generally, you see a lot of cancers that is actually coming from the skin, right? Or let's just say gut epithelium, right? Or bone marrow, right? So this is something that you have to remember. And remember always this genetic alteration things. Now after this, in order to call these cells to be neoplastic, what are the features the cancer cells needs to have? That's what we're going to talk about. Remember, there are some hallmarks of the cancer cells, and that you need to remember this, right? So let's talk about what are these hallmarks of the cancer cells. Let's just say hallmarks of cancer cells. And we'll talk about what this means. Look, in order to be cancer cells, right? First of all, one of the things that really happens is that, after gaining, let's say, after basically, going to the genetic instability, right? And because of the mutations, basically what really happens is that for one of the first thing they do is, what do they do? They have something called they're autonomous. Let's write down here. They're always a hallmark, autonomous. You need to remember this. And what is autonomous basically means? Well, autonomous growth, that's what we're talking about, which means that they're self-sufficient growth, right? Meaning this, that, they do not require any sort of growth factors or any stimulus for them to actually grow. They automatically, they actually grow. And even if you actually remove or stop the stimulus, the primary stimulus, they still grow, right? So that's what happens. The autonomous growth, which basically leads into self-sufficient growth, that's what this autonomous growth means. And this is one of the hallmark of cancer cells. Second, you need to remember, is that, remember how we said, they're insensitive. Let's just say, they're insensitive to basically growth factor inhibitors, okay, or inhibitors. They do not respond to any sort of like signal mechanisms, for them to stop. Like for example, if you give, let's just say, if you try to stop from the cell to proliferate, overproliferations or overgrowth of the cells, they're insensitive to that, right? That's what happens. Third, not only this, remember, they evade, as we said, remember, they evade what? ATP dependent what? ATP dependent programmed cell death. This is very important. This is one of the features of that. Fourth, not only that they will have evade ATP dependent programmed cell death, we'll talk more about what this all means, right? They also have what? Remember, they also do something called, look, they upregulate. Let's just say they upregulate telomerase enzymes. What does this means? Look, you know what telomeres are in DNA? Like for example, let's just if I said DNA, at the end of this, like let's just say chromosomes, you have a telomeres, right? And the telomeres are basic the nucleotide sequence, right? And as every time when the cells multiply, right, this DNA gets shorter. That's what happens. And basically, the cell can actually divide up to maybe 40 to, let's just say, 60 times, about that, okay? Normal cells. And after that, cell can no longer go through a process of mitosis or cellular divisions. But what happens with the cancer cells is they upregulate this enzyme called telomerase. So every time when the DNA gets shorter, right, because of this telomerase, they add a nucleotides, new nucleotides or additional nucleotides, and it's because of that what happens is that they don't get shorter, right? That means that they are always going through a process of cellular divisions, right? So there's an unlimited cellular division happens because of this telomerase enzymes, right? It's because of that these cancer cells usually become undying, or let's just say never ending, or they basically becomes immortal. Very, very important, right? They become immortal, right? That's one of the features of the cancer cells. And not only this, look, what are the things that happens with the cancer cells? Look, when this upregulation telomerase enzyme is there, right? And there's unlimited cellular divisions is there, that basically causes the cells to what? Cells to over survival of the cells or over proliferations of the cell. That's what happens, right? Now, 5 is that, look, they also sustain what? Angiogenesis. Angiogenesis, meaning that, they are important for basically forming a new blood vessels. Because remember, the cancer cells requires nutrients and also blood, oxygen, other different nutrients for them to actually, proliferate and to over survive, right? So this is what they do, sustain. 6 is very important too. What happens is that after sustaining engine, they also can invade and maybe let's just say metastasize to, let's just say, maybe lymph node or maybe other tissues in your body. They can invade and they can metastasize to other parts of the body. They have the ability of, invading and moving to other tissues. That's what they do. That's a 7. Look, they can actually alter the metabolisms. Okay, they alter the cellular. Basically, they can alter the metabolisms, normal metabolism. What does this mean, though? I'll talk more about this when we talk about here. All right, just hold on here. And then 8. Look, they can also evade what? Evade from the immune systems. They can also evade from the immune systems. Like they can basically suppress the immune response, so that way they don't get attacked by the immune cells, so they don't die, right? So these are the hallmarks of the cancer cells, right? Now you need to remember, again, whenever we talk about the neoplasia, right? Neoplasia is basically what? It is a new growth. It is an abnormal growth. Look, so let's write here. It is a neoplasia, which is an abnormal. We can also say this is an abnormal mass of tissues, okay? Mass of tissues, right? Which has the properties of multiplying, uncoordinated, unregulated cell division. It can go. It can evade self-programmed death, right? It can invade. They can do that. And the growth is what? It's excessive growth. It's uncoordinated growth, and it's over proliferations of the cells. All right. So this is what usually the characteristics of neoplasia. So by definitions, whenever you want to explain neoplasia, is you need to remember that neoplasia is basically a new growth, but it's abnormal growth of the tissue which grow autonomously, right? It is self-sufficient growth. It has a property of growing excessively, and it goes persistently grows, and it evade apoptosis, right? So these are, and all these other things if you want to mention too, but you need to be very, very clear about what neoplasia basically means, all right? Now let's talk about how these are the changes you can see in, let's say, these cells. Like for example, this is a cell right here, right? This is a normal cell, right? And what happens is that, let's just say, for example, if you get a wound, if you get a cut, right? If you get a cut, then what happens is that when you get a wound, there is a cut is there, and your cells have to proliferate and actually fill that wound, right? Now, when that happens, usually, look, let's just say there's a small cut here, right? The cells will basically come and multiply and basically, fill this wound, right? That's what happens. But remember, and as this filling that wound, really with the cells, what happens is that there is a property of something called contact cells, right? The cells that are communicating, so that way the cells prevent from actually proliferating overly, right? In normal cells, right? If there's something has happened, there's something caught is there, your cells don't keep multiplying or proliferating, right? There is a stop. So cells are usually well organized and well regulated and well controlled by our genes in our body. Every cell has about 30 genes, and these genes have different functions, right? These genes are called, let's just say, they are called the protogenes, right? The protogenes, and these protogenes are basically, they make these proteins, and these proteins have a functions which basically result into what? Which basically result into, maintaining the normal architecture of the cells, and also they controlled our basically cell cycle. That's what they do. But whenever any defects happens in our genomic instability happens, or genomic alterations happens, when we lose the functions of the cells that basically maintain the normal architectures or the well-controlled properties, then basically that's how it leads into what? The neoplastic. It can transform into neoplasia. That's what usually happens, right? So cells ways usually, when cells communicate with each other, sometimes there's a process of called contact inhibition. That's what usually happens, right? And then cells no longer are prevent from overgrowing or overgrowth of the cells, right? But let's just say that this cells right here, this is my one cell right here, right? Let's say these cells basically went through a process of mutations, right? Remember, whenever cells go to, there's something called proto-oncogene is there. What are proto-oncogenes? Basically, these are inactive form of the oncogenes. Oncogenes is referring as the cancer genes, all right? So basically, this is an inactive form of the proto-oncogene. Whenever, let's just say, there is a mutations happens, right? That result into what? Oncogenes, right? So that basically result into what? Oncogene, which means that the mutation is to happen. But what kind of mutation happens in proto-oncogenes? There has to be maybe like a gain of function mutations happens. All right. So whenever there's a gain of function mutations happens, okay, or there's a translocations of the proto-oncogenes happens, then basically that result into what? That basically result into over giving like signals, which basically result into over proliferations of the cell. That's what really happens, right? So let's say when the cells, let's say these cells went through a neoplastic transformed, okay? Let's say it acquired mutations, right? When it acquired mutations, it basically resulted into what? Over proliferation of the cells. So let's just say these cells started over proliferating. Let's just say like this, okay? Over proliferating, and it's gonna do what? It's basically gonna make daughter cells, right? Remember, almost all the time, the cancer cells actually originate from these cells, which is one mutated cells, right? And then it started making the daughter cells, and these daughter cells are gonna make like other daughter cells like this, right? So it's basically making a mass of this tissue like this, all right? Now, what is happening here is that, well, there's a nucleus is there too. That means that all of them have what? This cell went through mutations that basically mutated two daughter cells, right? And those two daughter cells pass, or they made another like 4 daughter cells, then 8, like, that's how it goes. And basically, all of them have what? Mutated, okay? All of them are mutated. All of them are mutated cells. And because it actually originated from one cell, this one, okay, and they make a lot of progeny of this, right? They make the clone of these cells, and they all have the same mutations. We call this concept monoclonal, right? This is what monoclonal basically means, because this is a mono from cells one mutated, and they give all the progeny of the cells, right? This is what happens. Now, when the cells happens, when this is multiplying, and this making the abnormal mass, which is a transformed neoplastic, this is what happens. This is basically leading to what? Hyperplasia of the cell. That's what is happening here, right? Now, this is what happens. Now, after that, as cells keep multiplying and multiplying, look, right, like this, right? This basically gonna go a process of what? Let me erase this. This basically gonna go a process of here, right here. This process is going to go what? Normal cells, they are mutated. That went to hyperplasia. Let's just write on it. First, it went to hyperplasia. That's what happens, right? Then what happens? Then after that, what happens? Slowly, it can lead into dysplasia. That's what happens. Now, the concept is, what is dysplasia? You have to remember this. Okay, this is a series of things that's going to happen. Which means that, look, number one, you basically this cell, it's going to lose the uniformity of the cell size and shape, right? Or simply we can call them a pleomorphism. That's what happens, right? That's what happens. They're going to lose the cell size and shape. That's what they're going to happen. Second thing, that's what is going to happen here is that, look, they're going to lose the architecture. Look, it's normal like this. It lost the architecture of the cell. That's what's going to happen, right? Lost the architectures of the cell. That's what's going to happen. In the dysplasia, third, the nucleus here, the nucleus this nucleus, they started getting what? Bigger, right? So basically there's increased amount of DNA, right? That's what's going to happen. So you see the increased nucleus and then cytoplasm ratio. That's what you get to see. Remember, all of these things, dysplasia, it can be reversible, all right? Dysplasia. But there is another form which is called dysplasia in situ. Okay, or let's just say, instead of saying dysplasia in situ, which is actually a severe form of dysplasia, we actually call them a carcinoma of situ. What does this mean? we'll talk about what does that mean. Look, in the carcinoma situ, okay, this is a severe form of dysplasia, which means that look, there's entire thickness of this epithelium. That's what it happens. Okay, there's very, very entire thickness of the epithelium. But remember, one of the things very, very important with the carcinocytoma, or we also call them, let's say, pre-invasive. We can call them a pre-invasive too, right? In here, even though there is entire, let's just say, entire thickness, right? Entire thickness of the epithelium. What really happens is that, look, this green is actually basement membrane. It hasn't invaded the basement membrane. This is very, very important. There's entire thickness after dysplasia, which is irreversible. It can go another phase, which is called carcinoma situ. There's entire thickness of this epithelium happens, but there's still intact basement membrane, which means that it hasn't invaded the basement membrane yet, right? But remember, this form is called carcinoma of situ. But this carcinoma situ, it's irreversible. Okay, this is irreversible. So this is something you have to remember. And after this, what happens is look, it started invading, look, the basement membranes, and not only that, it can also invade to basically, let's just say if I made a blood vessels there, let's just say there's a blood vessels, all right? It can invade to the veins and all, right? Now, if this property is there, then we call them invasive, okay? When it started penetrating the basement membranes and also penetrating to like the veins, or let's just say arteries or lymph nodes, right? Then we call them invasive carcinoma. But then after that, what? Then we can even simply say, then later what happens is that, maybe let's just say that this little mass, right? It got detached, right? It got detached, and then it went to, let's just say, the liver cells, right? It went to the liver cells, and then it started making a mass here, right? And they started growing here, right? Now, whenever this transformed neoplastic cells, it got detached, right? And basically through this vein, let's say portal vein, it went to liver cells, and then they were homing in the liver, and they started replicating, and they're making another abnormal mass there. Then we call them a metastasis. Now these cancer cells have been metastasized. Okay? Now, again, see, these are changes that you see in normally, all right? It's basically one transform cell. There's a monoclonal proliferations happens, right? Basically leads to different series of cells of hyperplasia, then it's going to lead to dysplasia, right? And then basically carcinoma situ, and you're going to have invasive carcinomas, and then obviously the cell can also metastasize, right? These are the things that happens. Now, I'm going to actually quickly show you the picture actually. I got this from the first aid book. And basically if you see right here, like, this number one is actually, it's basically showing a normal architectures of the cells, like with epithelial layers, and you can actually read the basement membranes and the blood vessels, right? But if you look at the number two here, look at this. You see up there is a nuclear changes, and you also see the cell size and shape has been changed here, right? And this is basically showing you the dysplastic cells, right? And this is the series of changes that you see in the cancer progressions, right? The tumor progressions. And if you look at the number three, this is where we're talking about the carcinoma situ. Remember that there's intact basement membrane here. It hasn't penetrated the basement membrane, and that's what you see in the carcinoma in situ, right? There's entire thickness of epithelium without penetrations of the basement membrane. And the four is showing there is an invasion that actually invaded the basement membrane by what happens is that this cancer cells actually secretes the enzymes like collagenase enzymes or metalloproteinesis enzyme, which basically breaks down those basement membranes, right? And they start invading the deeper layers. And then if you look at the number five, and here you basically see that there's also invasions and then spread to the distant organs like via the lymphatics or the blood vessel. That's what this number five is showing, right? So this is a series of progression that you see it. And if you look at it here, if you look at, let's just look at this one right here. Look here, this is basically showing the normal cells right here. It is not, if you look at the nucleus right here, it's not as dark as compared to this one right here. This is basically showing carcinoma of situ, like, basically the tumor cells actually accumulate abundance amount of the DNA, and then when you stain DNA, it appears more in darker, right? That is why it appears more in what? Hyperchromatic nuclei. That's what you get to see right here. And this is basically showing the carcinoma site right here. This is like basically maybe moderate versions of dysplasia. This is like maybe mild. I started getting this dysplasia right here. All right. Now let's talk a little bit about that. What is all this? What is like generally this evade of ATP dependent programmed cell death? What is all this means? We'll talk more about that. But before we go over there, right? One of the things that you have to remember here is that these cancer cells, right? Whenever there's the cancer cells is there, right? The cancer cells, even though when there's a lot of multiplications of the cells happen or proliferations of these cancer cells happens, one of the things that happens is that, in order for them to survive, right, what do they need? They need energy, right? Or they need different carbon materials to maintain the abnormal cells, right? And how do they actually maintain and how do they actually over survive? That's what we talked about. Now, before we talk about that, let's talk about two concepts here. Usually in the cancer cells, right, there are compositions of the cancer cells. You probably have heard about let's just say the parenchyma and then stroma. Now let's talk about what does that mean. Those are the compositions of the cancer cells, right? So let's write down here. Here, okay, we'll do compositions of the cancer cells. Let's just say compositions of neoplasia, right? And usually in this care, we have to remember parenchyma, okay? Parenchyma, and you have to talk about stroma. Let's just say that there is the cancer cells of your, let's just say, skin, okay? This is skin right here. And this is look, if this cell is going to transform cells, right? And start proliferating like this, right? And it basically making the, what? Abnormal mass, which has the properties of all of this, right? That's what's happening. But remember, it is localized, okay? It is actually located only in the skin. It doesn't have a property of, like, let's say, it hasn't invaded yet, okay? It hasn't metastasized yet, and it hasn't acquired new blood vessels or connective tissue yet, right? If that is the case, okay, there's a localized abnormal mass is there, all right? Let's try it out here. There's a transformed neoplastic is there, neoplastic cells is there, all right? And basically, abnormal, localized, innocent mass is there, right? Then we call them a parenchyma, all right? But let's just say, again, what's the same thing here? You have a skin right here, right? But these cells right here, there is this abnormal mass is there, right? This mass, this is abnormal. But on top of this, they started acquiring like blood vessels there. There's a lot of these blood vessels out there, right? On top of this, there is also, there's also what? Fibroblast cells out there, right? There's a mesenchymal cells out there, like fibroblasts or like collagens are deposited. Let's just write down here, collagens, right? Collagens. This cancer cells started producing this fibroblast cells, and there's a lot of these collagen cells, and the fibroblasts are there. Then, all right, different connective tissues plus like blood vessels are there. Then we call them a stroma. Okay. Now, next time, you don't want to get confused with parenchyma and stroma. It is very, very important. These are compositions of the cancer cells, all right? And remember, this parenchyma, what is the importance of parenchyma? Remember, parenchyma basically help us to what? Determine the names of the tumor, okay? Whether it is, let's just say, whether it's a epithelium in origin, or let's just say the mesenchymal origin, right? It can tell you. And it can also tell us the biological behavior of this cancer cell, whether it's going to what? It's going to rapidly grow, whether it's going to slowly grow. It's very important for prognostic factor, right? So basically, look, it can tell us the names, like help us do classifications of the tumors, plus it can also help us to do what? Behavior, biological behavior of this transformed cells, right? It is very, very important. When you talk about stroma has basically what? Now they acquired new blood vessels, right? That's what they're going to sustain their blood vessels. On top of that, they're also acquiring what? The new connective tissues. They have fibroblast cells. They're matrix. They have collagens out there, right? And remember, there are different amount of, let's just say this collagens, that determines whether they're going to be a soft and flashy tumors or they're going to be shard, solid or hard or scurous tumor. Let's just say that if there is let's just say new blood vessel but relatively less amount of connective tissues, then your tumors can be like soft and flashy. Like that's what you get to see in one of those tumors in breast sometimes. Okay, or let's just say that if there is a lot of these tumor cells acquired a lot of this collagen, lots of collagens, and there's it becomes hard. Okay, it becomes hard. Then we call them a what? Desmoplasia reaction. Okay, so what is the desmoplasia reaction is? Usually desmoplasia reaction basically means that these cancer cells have acquired like a lot of this matrix, and a lot of collagens are actually deposited, and these fibroblast cells are all deposited within the tumor, right? And the tumor becomes very solid and hard, right? And that is called desmoplasia reactions, or we can simply call them a desmoplasia. And sometimes what also happens is that these tumors will get basically get hyalinized and have a lot of this. So much of collagens are deposited there, it basically becomes very hard, and we call them, and it appears as scurous, and we call them a scurous tumor. Let's just write it. SCHRRIOUS, scurous. Okay, so this is also something that you remember. Remember, usually this is the difference between parenchyma. Parenchyma is a new transformed neoplastic cells, which basically help us to classify the names of the tumor, plus it can also tell us the biological behavior of the tumors. Whenever we talk about the stroma, they contain what? They acquire new blood vessels, plus there's new connective tissues out there, right? It's the amount of the connective tissues basically determines whether the tumors are hard like scurous, or let's just say the tumors is soft or flashy, right? And remember, these are usually the compositions of neoplasia, right? This is something that you have to remember. Now after this, let's go and talk about, there's a concept called Warburg effect. I have to talk about this. Okay, it's called Warburg effect. What does this mean? All right, and this is for every cancer cells. Really what happens is that whenever there's a cancer cells is there, right? Cancer cells basically, one thing they do is that they basically induce something called hypoxic induce factor gene 1, okay? It's called hypoxic factor inducible gene 1, right? And one of the things that it does is this hypoxic factor inducible gene is that, they can actually cause the overexpressions of this transporter called GLUT1 transporter, right? Let's just say GLUT1 transporter. And what is the importance of the GLUT1 transporter? Whenever there is a GLUT1 transporter is there, right? What is going to happen here is that they can take a lot of, this is a transport, lot of, what glucose. And glucose is what? 1, 2, 3, 4, 5, 6. It's a 6 carbon molecules, right? Glucose. And this glucose, right, what happens? They can actually run the glycolysis, right? They can do that. And when you run the glycolysis, you make pyruvate, right? That's what you make. And from the pyruvate, you have two ways you can do. You can make here, this is, let's just say, it can go to the TCA cycle, mitochondria, and basically go oxidative phosphorylations, or it can go to lactate. Look, it can basically make lactate. Look, right? This is what usually happens. Cancer cells, what they do is that, usually, even in the presence of oxygen, because of this, right? Because they induce this factor, they take a lot of glucose, right? And I'll tell you what importance of glucose. And they take a lot of glucose. But even in the presence of glucose, they do not go to the pathway of this. Even in the presence of oxygen right here, they do not. Normally what happens, you run the glycolysis, you make pyruvate, and you go to TCA cycle, and then from there you go to mitochondria. This is mitochondria, right? And you do oxidative phosphorylations and you make more ATP. That's what normally you do, right? But usually in the cancer cell, what happens is that this pathway is sort of inhibited, right? They pass, and what happens is a lot of pyruvate they make, and they make, like what happens, this pathway, they actually make a lot of this lactate. That's what it happens, right? So there's basically, less oxidative phosphorylations happens, right? This whole phenomena of where the cancer cells actually take a lot of glucose. There's a glucose there. These cancer cells are like hungry for glucose. So they take a lot of glucose and they basically run the glycolysis. They make pyruvate, and then from the pyruvate they make lactate. And then they use lactate as their energy source. That's what they do, right? And remember, they use this lactate as energy. This phenomenon is what is called Warburg effect, right? And what is the importance of this taking a lot of glucose in the cancer cells is that, we use this for the diagnostic method, okay? And what is the diagnostic method? For PET scan. Like, think about it. In the PET scan, like, what happens? In PET scan, you basically do what? You basically give a radio-labeled glucose. That's what you do, right? You radio-label glucose, which is non-metabolized by the cancer cells, right? That's what happens. So what happens is when a lot of these cancer cells actually takes the glucose, right? This radio-labeled glucose, right? And basically when you do this PET scan, you basically see those lights, and the amount of glucose, because they're hungry for glucose, right? And that is the importance of this whole concept of this Warburg effect. And this is very, very important. You have to remember this, that, there's overexpression of GLUT1 transporters, and they take a lot of glucose, right, by the cancer cells. That is the reason why, the PET scans are very important, and we're basically looking at the, how much glucose is taken by the cancer cell. That's what's happening, right? Here, I'm actually showing you the image of the PET scan, and I got this image from this site right down here. So if you look at it, that the cancer cells clearly takes a lot of glucose, right? So it's because of that, when you do a PET scan, it will light up, it will show this, right? Now let how I said that there's a lot of this lactate is there, right? Whenever this lot of lactate is there, what is going to happen? Think about this, right? Look, this lactate can actually come to the what? Circulations, right? And when the lactate doesn't come to circulate, what happens? That can actually lowers the what? pH. It causes what? Metabolic acidosis, right? It can cause acidosis. And remember, and when there's acidosis is there, that can actually what? They can actually impair the functions of our different enzymes in our human body. That's what they do. On top of that, they basically what alter the metabolisms of our cells. That's what they do. And on top of that, whenever there's a lot of acidosis conditions, what is going to happen? Remember, this suppresses the immune system as well. So this is, look at number 8. Look, invade, alters the metabolisms, or evade from the immune system. This is one of the way they can actually evade from the immune systems, and also alter the normal metabolisms of the cells. That's what they do. All right. Now again, look, whenever they're doing all of this, right? Not only this is happening in here, they're also changing. Look, when they're making a lot of lactate, right? There is a oxidative phosphorylation, which is probably happening less or not at all. But remember here, there's still presence of, their oxygen is there though. In spite of presence of oxygen, they still, go and produces lactate, okay? And basically, this whole phenomenon, it's aerobic glycolysis, right? That's what basically happening. Even with the presence of oxygen, they're still making lactate, right? That's one of the key stuff or key concept that, that is explained by this Warburg effect, all right? Now, let's just say that, when there's a cancer cell, when the transformed cells happens, right? What are the changes that could happen in, let's just say, in cell? Remember, what happens to mitochondria? Let's just say, remember, you're making less ATP, right? So what happens? Mitochondria is start getting swollen. That's what's going to happen, right? Your rough endoplasmic reticulums and all, what is going to happen? They're going to increase in the number of rough endoplasmic reticulums or ribosome. They started increasing because remember, they start making a lot of these oncogene proteins. That's what they're going to start doing, right? And not only this, they can also change the cytoskeletons of these cells, normal cells, right? That's what they do. And this plasma membranes, there's also, you see alterations in plasma membrane because remember, they're making the oncoproteins, they're making your cancer cells proteins, right? So they're basically going to compromise the functions of the cells by bringing different alterations in the cells, right? That's what they're going to do. And this is something that you have to really remember. I'm going to quickly review, okay, of what we talked about so far. Remember, in order to be cancer cells, right, it has to be unregulated, as we talked about more, right? Unregulated, excessive, persistent growth, it has to be autonomous, right? Self-sufficient growth, it has to be. And remember that, it needs to evade ATP, let's just say ATP-dependent cell death. And how do they do it? One of the things they do is, look, when there's a series of mutations that happens, right? One of the things they can do is that they can actually suppress this, they can suppress the tumor suppressor genes. That's what they do, right? And when you suppress the tumor suppressor gene, what happens? That basically results into what? Over survival of the cells, right? Remember, the cells, they're usually normally, let's just say if this is a cell, right? Let's just say this cell is defective, right? Let's say this cell is defective. And normally what happens is that if these cells, there is a system, there is a basically self-sensor. This cell itself has its own sensor. If it's damaged or if it's defect, it will sense by its own cell, and it will commit suicide, right? That's what is called apoptosis. But then what happens with the cancer cells is that, instead of committing suicide, they started what? They basically gain the tumor suppressor gene that actually favors what? Favors over survival of the cells, right? So basically what they do is that, there is a pro-apoptotic genes, and then basically there's anti-apoptotic genes. So the pro-apoptotic gene means which favors the cells to commit suicide, right? There will be loss of function mutations of the pro-apoptotic genes, and there is a basically gain the function mutation of the anti-apoptotic gene, okay? And when there's a more level of, there is a ratio of the anti-apoptotic with the pro-apoptotic, the cells will what? Over survive. That's what happens, right? Remember, by having an autonomous growth, self-sufficient growth, and insensitive to the growth factors and inhibitors, right? That basically gives what? Basically causes cells to over-proliferate. Cells is going to over-proliferate like this, evading from the ATP, right? And then upregulating telomerase, right? This basically leads into what? Over-survival of the cells, right? That's over-survival. And by giving sustained angiogenesis with the new blood vessels, then more glucose is going, more nutrients are going, right? And the cells requires that, right? And that's what things that happens. And then obviously they can metastasize to the invasions and go to lymph nodes and other tissue. That's what happens. And then as we talked about how it can lead into altered metabolism, evade system, how it evade from the immune system, which I talked about it, right? Now after this, let me quickly talk about how usually the clinicians, or let's say pathologists, how do they actually, grade or stages this tumor. There's a whole concept of grading tumors and this stage this tumor. Let's talk about that quickly, all right? Usually it's pathologists, right? Let me write it down here. Usually pathologists do something called grade. Okay? They can grade tumors by basically saying 1, 2, 3, and then 4. This is how pathologists can grade the tumor. And the grading tumors is based on degree of cellular differentiations and the mitosis activity. Very important concept you have to remember, okay? Degree of, let's just say, degree of cellular differentiations, all right? And second is the mitosis activity, right? So let's just say that, and remember, grading is always done by what? It's always done by pathologists. And how do you do grade? You have to do biopsy. It's a very, very important concept. Remember, it's a biopsy is the key, all right? So you do the biopsy of this, all right? Now, you take the biopsy and you send it to the pathology lab, and basically they'll look at it, and if the cells are, let's just say, well differentiated, all right? If the cells are well differentiated, then what happens? Usually those are low-grade tumor, okay? But let's just say the cells are undifferentiated, or let's say they're anaplastic. Remember, anaplastic, you never want to have anaplastic cells, right? Because these are undifferentiated cells, right? So usually those are more aggressive, all right? So that's how the grading can work. Not only grading they do, but there's also something called stages, okay? And what is the difference between grading and stages? Grading you do with biopsy. Stages is you're done by maybe radiology or through imaging, right? Radiology is important, or let's just say through imaging is also. And for this one, you have to remember this concept, TEM. What is TEM? Okay, so not TEM, it's TNM, sorry. It's TNM. T is referring as a, this is how they actually, stays the tumor. T is referring as a tumor, like size, okay? This T is referring as a, let's write it down here. T is referring as a tumor size, all right? So let's just say the T is referring as you have a tumor size, all right? And you can say like, okay, T1, T2, T3, and T4. And there is N is referring as a nodal involvements, meaning that whether or not it has spread to the regional lymph nodes, all right? And you can simply say this is maybe N0, maybe N1, N2, N3, okay? And this M is referring as whether or not cells has been metastasized or not. You can do M0 or M1, okay? So what does this mean? So whenever we say like tumor size is based on okay? This is how the clinician can use, right? This is radiology. So basically, let's just say, if I say, let's just say this, the cancer cell, this transform is, let's just say T3, and we can simply say this is, let's say N2, and we can simply say M0. If we say that, that means that, look, T3 is that means that the size is relatively big. It has, N2 is referring as it has involved nodal involvement, but M0 is referring as it hasn't metastasized yet. But if I change this and let's just say, if I say, okay, there is a T3, but then let's just say N0 and M0. If I say that, that means that there's a tumor size, there's a large tumor cells is there, but it hasn't involved, N0 is referring as it has not involved no lymph nodes yet, and it hasn't metastasized yet. So whenever, if you use ever, like, let's just say if they say grade 3 tumors, that means that you're looking at if the tumor is a grade 3, then you're talking about the cells is what? Cells is basically, look, if it's a grade 3, that means cells are probably atypia, right? Cells is atypia. It has what? It has already go to like mitosis activity. It probably have endothelial proliferations, and sometimes maybe necrosis is there too, right? So basically, it tells you about what? It basically, grading tells you about the mitosis activity and also these cellular differentiations of the transform or the neoplastic cells, right? And you always do biopsy to determine that. And only pathologists do that, right? And only pathologists can do that. With the stages, usually by the imaging, you can actually basically label each one of them by T and M, which basically tells you the tumor size, and N is referring as whether or not it has involved nodes, and M is basically referring, as I said, as the metastasized. All right, guys. So, again, this is all I have, guys, for you today. And next class, we'll talk about the nomenclatures of the tumors. All right? Thank you.